Okay, so please, would you be so kind as to sit? Ricardo. So the Spanish team is okay? It's fine now. Okay, so please take your seats. Ricardo, Philippe, Jean-Pierre. Shall I use the name and shame? Okay, so we continue with the second session, still on pest and disease monitoring. And so Ellen Pelgrims from the Flemish, one of the, well, we are two, by the way, we are two funders in Flanders, and she's going to chair this second part of the session. I'm just chatting, time that you take your seat and you become silent. Yeah, it's coming now. Okay, now the floor is yours. Okay. Thank you.
Good afternoon, everyone. I'm Ellen Pelgrims. Antoine already said it. From the Flemish Agency for Innovation and Entrepreneurship. I have the honor to guide you to the hardest part of this day, meaning the last part. And I hope you have all enough energy to listen to the last 4 speakers. There is a minor change in the program. We start with Didier Andrivon with the project MLight 2.0. So where is, okay. So.
How does it work? I don't know. Okay, yeah. So, hello everyone. So I'm leading the project IPM MLight 2.0. And this project was funded on the first call. So we started actually in April 2016, and we will finish early next spring. No, it shouldn't be the right direction. Okay, so the project deals with late blight on potato. If you ever grown potatoes in your home garden, you know the disease. It's a destructive pathogen. It's very costly because the cost of fungicides and losses have been estimated over 1 billion euros a year for Europe only. And it's a re-emerging pathogen worldwide. The pathogen is incredibly diverse. Here you've got a map from the Européen network, which is a European network dealing with blight control. And on this map, so that's for 2014, but we have about the same picture every year. You can see every spot is a different isolate or different sample. The colors correspond to clonal lineages, so genetically related genotypes, and so those of the same color are actually a single genetic unit, sort of. You can see on that map that some of those clones are very widespread. It's the case of the blue dots that go all the way from the UK to Russia. They're also present in other parts of the world. Some are more localized, like the green ones in France and Ireland. And you see also a lot of gray ones, which are unique genotypes, which probably sign sexually reproducing populations, with lots of recombination there. Those populations are not stable at all. And if you have a follow-up of those populations over time, you can see very rapid changes and very unpredictable changes. I don't know if you would have been able to say, to guess the distribution in 2007 from the one in 2006, but I wouldn't. And you see, we changed the red ones by the blue ones and pink ones, etc. And nobody makes sense right now of the timing and the reasons for those changes. I said those lineages are actually groups of genotypes related. But within those lineages, there is a lot of diversity. There are still subclonal variants that appear and that are dispersed over a very large phylogenetic tree. So it means that within those lineages, we expect a lot of variation also for phenotypic traits, which are the ones that are of interest for us for the control. But we don't know the distribution of those phenotypic traits across genetic variants. What we know is that variation occurs for many different phenotypic traits. Here I've got an example with virulence, so the ability of overcoming resistant genes in the host. And you see that within the blue lineage or the pink lineage, there is a lot of polymorphism or polyphenism for virulence. You've got a very fluctuating frequency of overcoming and non-overcoming, virulent and avirulent individuals for every single gene. So it means that if you have the identification of a genotype or a clonal lineage, it's still very difficult to make an inference about which genes will be useful or not. Sorry. Another issue is that all those lineages do not respond equally to climate. Those are data that are from a PhD that was made in my lab some years ago. And you see that there are significant differences between the pink and the blue lineage in median growth rate or spore production or spore size according to temperature. But all the decision support systems are tailored over a uniform response to climate, which means that if you have a population that is made predominantly of the pink one, the pink lineage, or the blue lineage, or even other lineages, they are assumed to react the same to temperature and humidity, which is obviously not the case. So you might have problems with a correct risk prediction because of that variation. The good news is that the nasty ones don't ever win. Here you've got a very short chart about the transition between the 2A1 to the 13A2 lineages in Europe, and the normal guess would have been that the reason for that is that the 13A2s would be more pathogenic. That's because we are pathologists, you know. So we always look for the worst. But indeed, if you measure the pathogenicity of those lineages relative one to the other, you actually realize that the invader is actually the least pathogenic of the three. So it doesn't make sense, really. Well, it does, but... So when we started that project, we were able to quite accurately describe the situation and explain the changes in the past. We still had some tools to follow current evolutions with the markers, the genetic markers we had, and also with the phenotypic tests, biotests we had at hand. But we still had a very hard time, and will still do, predicting future changes. And that's actually what's important for the grower, because when they decide which variety to grow, when to spray, what kind of practice you should have on your crop, you need to know what kind of pathogen you're targeting. And the second issue was that, as I said, the population data are currently completely ignored in the decision support systems, which are based on climate only. So, because of those concerns, the Euroblight group had a statement back in 2015 during its meeting in Romania, and there were 4 points there. We need to monitor the population. We need to have a better link from genotypes to phenotypes. We need to have DSS systems that will take that information on board, and we need international collaboration because obviously the pathogen doesn't stop at borders. And that's actually what we made when we designed IPM Blight 2.0. You find the 3 work packages with the sampling and phenotyping, population, the phenotyping, the DSSs, and the international collaboration. So we actually just matched the recommendations of Européen. Just a word on why it's called IPM Blight 2.0. It's based on the IPM 2.0 concept that was published a few years ago by colleagues in the Netherlands. The idea is that we need to combine all available information to have pathogen-informed strategies. So we need to monitor the population. We need to have a phenotypic assessment of what they do. We need fungicide anti-resistance strategies, but we also need host resistance management strategies because resistance genes don't last forever. And we need simulations models for better DSSs. The project has several partners in 6 countries. And what we did actually, or some of the results we have, one of the aims was what Jens Hansen in Denmark calls hunting the new. He is very keen on finding new things rather than old ones. So the idea was that in the surveillance system, we would be looking for new emerging genotypes or pathogens. And we actually, I don't know whether we were fortunate or unfortunate, but we actually spotted 2 cases of new invasive clones in Europe during the project. One in already clonal population, and that's the case of the genotype that is called 37A2, and another that invades predominantly sexual population, which is kind of surprising because those populations, because of their sexual status, are supposed to be more resilient to invasion. Okay, just to show you how it works. So that's 37A2. It started from one spot here in the north of the Netherlands in 2014. That's the first detection we have in our records. 2 years later, it already crossed the channel, spread all over Benelux to the northern France, also to some parts of eastern France, and some isolated spots here in Germany and in Austria. Oops. And 2017, you see that there was an intensification of the frequency of that genotype all over northwestern Europe. 41A2, it's a Nordic clone, I would say. It's the red one on those pictures. It was first detected in 2012 in Denmark, and now you see it has entirely replaced the blue 13 there. But it's also, if you look at the top graph here, sorry, at the top graph here, it has also replaced part of the sexual lineages or genotypes because the proportion of gray dots seems to be a little bit smaller now. So it seems that those populations are slowly converting to clones. Why is that? Again, we turn to phenotypes to try to explain that evolution. And we looked at fungicide sensitivity, at pathogenicity, aggressiveness, and the ability of overcoming resistant genes. If you look at those two clones, 37 and 41A2, you might note here that there is a quite large proportion of isolates within those clones that are quite resistant to a very widespread fungicide, which is fluazinum. So maybe that's one of the explanations. It's certainly not the only one, because there was already some variation for fluazinum sensitivity in pre-existing clones, so it doesn't explain 100% of the invasion success of those two ones, but it certainly has a role. And I have to mention that the development of 37A2s in Western Europe has almost eradicated the use of fluazinum in the UK over the last 2 years. Now, is it still the least nasty ones that are invasive? Well, actually, the 41 and 37 are not the most aggressive isolates here in that panel. They are least aggressive than, say, 6A1, which was pre-existing. So the trend continues. And if you look at 37, it doesn't produce that many spores either. So it's not like it's a super pathogen. It's just a super fit pathogen. Now, if I talked about intraclonal variation, that's the distribution of spore production of 5 clonal lineages, and each bar is a single isolate. They have been genotyped, they have been assigned to a lineage, and as you can see, you see a bunch of colors all over the place. So there is no sense there where you can say though the pink ones are more are producing more spores than the green ones, because there are also green individuals that produce a large number of spores. So there is no univocal relationship between genotypes and phenotypes. The same goes for virulence, for the ability of overcoming resistant genes. But it's still, there is still a trend that, if you look at the 41A2 here, it seems to be pretty nasty in the sense that it's able to overcome most of the resistant genes that are present in current potato varieties. We can also monitor that virulence directly in the field using film trap nurseries. So that's one such nursery where we plant differential varieties with different R genes, and we simply monitor the outbreaks and the dates of occurrence of the different the dates where those plants get infected and blighted. And what you get is, of course, there's a gradient in terms of the timing of the first infection. That's in Western France, that's in Norway, that's in Estonia, and that's in Denmark. But you can also see that within a single country that's not that big, actually, it's Denmark. If you do that experiment at 2 different locations, you get completely different kinetics of infection. So it means you need a local monitoring, not just a nationwide monitoring. The nation information is just not sufficient. The last step was to incorporate that information into better decision support systems. Again, that's the Danish system where you have the metadata, and they had recommended fungicide dose adjustments according to whether you have a susceptible variety or a more resistant one. Now, if we use the information for the trap nurseries as well, extra, we can even refine that recommendation. We can actually reduce the total amount of fungicide that is sprayed on the trials. As, here you see they went from 6.5 full dose sprays to a little bit more than 4, so it's a 33% reduction. Oops. With very good control. So adding the population information is actually added value. So some conclusions. Vigilance is essential, and we need to have that network. What's important is that it's not the same invaders everywhere. So you have to be aware and flexible to track those newcomers. It's also important to know the old ones because they are still there. As I showed, there is a very large subclonal variation, even in the new clones. And the genotypes with the markers we have now are not very good predictors of phenotypes. Oops. As I say, we can exploit that population information and with rather strong consequences. There are quite a few questions that are still pending, and that's why we are still going to work for the next 40 years or so. The question is, how far do we go with integrating that population knowledge into DSSs? That works well in experimental farms and experimental stations. Does a grower actually get the benefit of that information? That's still to be done. I showed you that the lock of the system is the phenotyping because it's a pain to do. It's just very time consuming. You need to get the isolates, you need to do the biotests. And usually you get the information 1 year after you sample the isolates, so it's just too late. So we need better tools, we need better biotests, but we also need better markers. And Antoine mentioned this in his introductory talk that actually the problem is getting multi-scale. Late blight is just one of the components of the fungal complex on the foliage of potatoes. The other main component is Antonaria, and it's increasing. It used to be controlled by late blight fungicide because they were very wide spectrum. Now we have more specialized products, so we dissociate the control of those two diseases. But the farmer doesn't care. I mean, he wants healthy potatoes. So we need to actually manage that as a blight complex. And that's it. Thank you very much for your attention.
So, are there some questions for now? We have the general discussion, but...
That's just a short leaflet we made for stakeholders. There are a few copies there for the ones who are interested, just to present it.
So, do we have a micro?
Who is collecting the samples all over Europe?
Oh, it's actually made. The question is who is collecting the samples all over Europe? It's actually an effort that is made through the Euroblight network. Euroblight involves all the stakeholders, including the chemical companies. They fund part of the genotyping. And the sampling is made actually on FTA cards, so it's sort of filter paper. You just print the symptoms, send that to the lab, and then we'll do the genotyping for you. The system currently collects over 1,000 isolates a year all over Europe. It's mainly coming from chemical companies, but also from advisory groups, from individual farmers. It's a big network.
Somebody
maybe I misunderstood. You said half the dosage you use. Did I understand correctly? What do you exactly mean? Because then I wonder how you think about development of fungicide resistance.
That's another question.
Yeah, because that's why usually it's fixed.
That's the Danish recommendation. The Danish recommendation is they have a limit on how much fung dose equivalents they can spray. So the strategy is rather to spray more often with lower dosage than to spray fung dose less often, because the pathogen is very epidemic. So that's why there are quite a few cases where half a dose is recommended rather than the full dose. That might be a reason for
me it's
for
a bit
emergence
surprising,
of,
because usually a product is authorized with a certain amount you are allowed to use per hectare and water volume. So I wonder. Well,
for what I know, but I wouldn't leave the floor to the Danish colleagues because I'm not the ones issuing those recommendations. It has been working quite well because we had preventative fungicides with large spectrum and very robust modes of action. Of course, if you go to very specific products with a single mode of action, the risk of fungicide resistance is much higher.
So maybe the last question and then...
Didier, I was wondering, is Phytophthora infestans also an airborne pathogen?
Oh, absolutely.
So is it more soil-borne or more airborne?
No, it's definitely airborne. If you look back at, I can refer you to the first epidemic back in 1845, where it spread about 500 kilometers within a single season in all directions from a single spot in Belgium. It's massive. But if you look at the maps for, say, for instance, 37A2 and the spread, it spread both east and west, and that's not through seed trade. So it's definitely airborne.
So because I asked you this question, for many diseases caused by soil-borne pathogens, soils, naturally resistant soils have been found, but this is not the case for...
It's not a big component in the system anyway.
Okay.
It might help prevent some tuber contamination, but it's not going to do anything to the epidemic development of the disease.
Okay, thank you.
Can I take this?
Okay. Very last question.
I'm just wondering about the resistant cultivars. Like, is that something that is going on, like with resistant potato cultivars for late blight?
There are a lot of new resistant cultivars that are being registered now. So there are more and more of them on the national list and on the European cultivar list. So you can buy them.
But is that like something that impact the DSS and?
Of course it does. Of course it does. The only problem that a lot of growers now spray their crops irrespective of the level of resistance the variety has. So they don't make full use of the resistance of the varieties. But there is scope for that.
Okay, thank you. Our next speaker is Ricardo Suey Cortes. He's going to tell us everything about smart DSS for IPM in protected horticulture. I think that.
So, good afternoon. I'm Ricardo Suey. I work for INRA at Sofia Antipolis, near Nice. and we work really in a decision support system. We are far away, not far away, but we are not so close to research as you do, as you will see. We are closer to information systems and to the application of this. We are really more in the development part of the situation. Just some part of the monitoring system has some special research, more in deep research, but the rest has been development, mainly development of the information system. So, as I showed you, usually this is what law asks for us, but this is what I would do. You see, it seems more information technology than agriculture. So what we have done is the problem. I have really followed the guidance of Laura, so it would be a much more flat presentation than the DDA one. So I think you can listen to me very easily because of my voice, or not?
For the record.
Ah, for the record.
Yeah, the movie stuff.
Sorry. I should. I didn't know that. Okay, so for the practical questions from the very beginning for us was that from many years ago, crops are subject to very strong market requirements, and this has led to intensive use of pesticides. This is the first problem, we all know. This is very general. And from the 50s, we are almost in a really what we call the classical all-chemical approach. Just we have a problem, we have a chemical treatment, okay? But this plant protection strategy, as you know, really exhibits very important limits nowadays, mainly because of the reduction of the available active ingredients. From what you know, all REAC and the diminution, the decreasing of all these active ingredients we are available now. And also the problems that we are facing now with the pest resistance. We are not talking about human health issues or environmental pollution issues, just talking about agriculture. We have these two ones. So in 2009, the European Union promoted the integrated pest management to be implemented as a crop production system. This means that IPM proposed the use of alternative approaches and techniques to chemical ones, and encouraging for sure the use of biological control agents when possible. However, what we have seen from that time where we try to implement this integrated pest management is that this is really difficult to implement and widely spread when we are talking at the plot level, at the farm level. So why? Because the IPM strategies are slow to take a sufficient robust concrete form to be really transferred to. I cannot stay there, sorry. But yeah. And also because this way of cropping, this kind of cropping system needs really a much better knowledge of the agroecosystem from the grower point of view. It's much more difficult to do it in this way than just to have the direct way of one problem, one pest, one treatment, okay? So, and there is also, from the 50s, we have 2-3 generations that are just doing this way. So we need also a change in paradigm, so the cultivation system and the grower's culture. It's not so easy to say, okay, from now onwards, you're going to use this IPM. So you need first training and to know better knowledge, and then also a change in mind in the culture of the society. So we think that the way to cope with these main difficulties, at the practical level, and when we say the practical level is at the growers, from the growers' point of view, is first to develop advanced tools for pest risk monitoring, because we have to know all the time what is the health status of our crop, and also to go with the second one, to assist them in the decision-making process. Otherwise, it's really important. It's really difficult for them. I came back. So what we are going to do, or what are we doing in this project? First, what we think they need, growers, to implement IPM is first sampling tools. So we have developed sampling methods and protocols to be easy applied and ready to use tools. Forecasting tools also to anticipate the population dynamic trends, also what we know now, what happened in our neighborhood, and what we can foresee that is going to happen in our crop in the near future. So then decision support tools, so to facilitate the decision making mainly from a global vision and a temporal vision of the agroecosystem of our crop. We also need to train them and to show them how to use it, so training tools, so to facilitate the knowledge of the agroecosystem. And finally, advisory tools, so with recommendation sheets and traceability of what we have done. So with this background, the general objective of Smart IPM is to develop, validate, and optimize a decision support system tool. But what it is very important for us is in a real context of greenhouse cropping season. All of we have done is done with growers in their own crops. So first validating that this is, they are using it, they understand it, and it's useful for them. Otherwise, we really know that it doesn't work. If we produce a very complicated, very nice decision support system from research, and it is probably the chances to be applied afterwards for growers are not so big. So one of our first points, or most important points, is to work in a real context with growers. So we don't start from nothing. I mean, we start from a scientific tool that already exists, a web-hosted database developed for experimentation in ornamentals, already at INRA in Sofia Antipolis. This is the new web address. And also, because we are working with protected cultivation, with protected tomato cultivation, we also start from all the knowledge we generated during the IPM Pure project for greenhouse tomato production in Europe. Okay? So we have a lot of knowledge already, and we have some kind of tool that we are going to adapt and make them use for growers. So for us, Smart IPM in the construction, it was important to have all the possibilities and all kinds of production of tomato. So from north to south in Europe, we have German partners, Spanish partners, and INRA at Sofia Antipolis. So we cover all Europe and all kinds of protect tomato production. Also, as I said, one of the main topics of our project was to our target group. Our target group, we really focus on advisory services and growers, because we want our DSEs to be used for them. Okay? And finally, these DSEs, as it has been built, is a link between all players of horticulture, because we are research, we have also experimentation units, and also the development and the crop production. So finally, it's a two-way link, a very good relationship. Providing the tools we are developing with them, methods for research, mainly for the monitoring protocols, and also the upwards, all the information we get from them, from the use of our system. Finally, this is very simple. Okay, our forward package is we focus first in sampling methods. We have to give them, if we think that monitoring is very important, we have to show them how to monitor and do it in an easy way first. We have to develop tools and methods, all the information system, what I mean. And then we have the testing of the decision support system in the different test growers. So, and finally, dissemination with training and all the information we give as today we can consider. At the beginning we thought it was a 30 months project, but this has really been a mess because we start our first protect tomato cultures in February, just when we know that the project was financed, was founded, and then but we could sign our agreements with the local funding agencies in June and September. So okay, our 30 months will finish in more than 3 years from the very first from our very first culture of tomato. So, and the general structure, so that's what it was. So 30 months was 2 iterative loops of development, test, validation, and then update of the system. So we thought to do it first on station and the second year on farm, but this was all mixed up, and we started in farms and station, and because we have the possibility to work with some growers from the very beginning, so we just do it. So talking about results, I'm just going to show three results. First, the web-based information system that you can access there. So just to mean that it's a reality, it's something that you can just check now if you have an internet connection. For sure, this is with a secure access. We had very important issues of security, traceability, and so on, and with also even more with the new law of information, personal information. And we start with a customizable dashboard where all the grower we need have the main information they need. This is customizable, so wow. They can put they are with a sorry, I don't see it. But okay, this is customizable. You can have all the information, the past information you want to be informed just when you connect. Also, you have there your program, your plan of new monitoring days, and so on, the climate conditions and other. Another thing that is very important is that it's adapted for us. It's adapted for tomato production. If we see that we are collaborating in other projects, so we are putting many money in this system with many other crops. You think that just do that only for tomato crops is, it's better to use it for many more of them. So adaptive in our case for tomato production, but we can have, and we have already many other ones. For example, you see, and is tomato production important over Europe? What does it mean? It means that we have for the Spanish advisors or grower, or for the German growers, this kind of different crop systems, they have different ways of looking at the problem. Mainly, this is related to the sampling protocols first, and then to the pest that we are considering, because we don't consider the same primary and secondary pest from the north and the south. So this is related to that, and also related to the way we are observing the pest. So are we counting? Are you doing a detailed or precise counting? Are we doing just a rough counting? Or we are just saying there is yes or not? Okay, this is dependent on the culture of everyone. Okay? So over Europe, as I say, because we have, for example, for the German case, we have 15 variables that they are considering to follow in a monitoring. And for example, in the Spanish grower one, we are just 10 variables following, okay, or monitoring. And also in user, because in Spain, we also work with some advisors, because the production, tomato production, mostly in Almeria, is very integrated. So in very big cooperatives, they have to follow some different protocols that they already have. So we have the possibility to customize this. So for users, we have the version for advisors and the version for growers. For sure, growers have more variables. It's much more easy for them. They really focus on primary pest. And also it's adapted for research. For example, this is the first year, what we say in research. We have more than 50 variables that we are monitoring in a tomato crop through advisors. So reduce to 15, just 15 variables, and also user, that we just keep 10 of them or less. Another result, this result is related to monitoring systems. Our colleagues from GKI, there has been a PhD thesis that has run at the same time than the project, and they have worked. They have an important problem with russet mite Aculops lycopersicii. So they have been working on new evaluation spectrum, new monitoring and pest detection methods. As you know, Aculops lycopersicii, we usually see it when the damages are very important and when these all spread in our crop. Okay, so I'm finished with this, and so they work in these two ways of early detection. So one with imaging, per spectral imaging, and another one with non-imaging with fluorescence. So they have had nice results, but you know, this is really from research still to work on that to be applied in real context. And also, for example, for them, many questions still stand and need more research. For example, for instance, is the time gain, because they gain just several days, enough to manage the tomato russet mite infestation spread or not? Because we have just 3-4 days that we are earning with these systems nowadays. So the transfer for us is warranted because we are working really in a participatory co-design approach. So growers and advisory services were integrated from the very beginning to define the needs. Has been training all during the project. Everyone that has been an update of the system, has been trained, and they have tested and used the system around. What it is, okay, the test has been made in a real context of greenhouse cropping systems. And as I say, the transfer for us is ensured through the prescribers and influencers, because this small quantity of people that has worked with us in Germany, France, and Spain, they are going to talk about it. And this is, for us, influencers is a very new word, very trendy nowadays, but we think this is one of the strong points to continue. For the development, the future in terms of the information technology system, information technology tools, you know, they are always in continuous development process, so we are not finishing there. But now we have translating the system in German and updating the English, Spanish, and Italian one. And we have a mobile application that we are already testing with beta testers. and we will have the final version for the end of the year, next month. And we are working also in a pest recognition module that will be integrated in the mobile application, also for the growers to easy the way to know to identify and recognize the pest, and also when they don't know it, just to take a picture and could send it directly to some advisor that could say what it is. And at the medium term, we are working. It's not that easy nowadays to connect the system to the plant protection bulletin, the French one from the beginning, to the phytopharmaceutic database. But we know that the databases that are already, they are not so updated, so we risk to have information that is several months, with a delay of several months of actuality. And we want also to integrate the network of climate, the weather station network, to have more climatic data. All the climatic data, the near weather station from the French network. So after we'll work in the other ones, but we are developing this here, and it's easier for us to work first with the French ones. So in terms of research perspective, we think that the early detection of pests and diseases, as they could be autonomous or automatic system, we know that and for us, this is our last word. Our idea is to have an important database of many growers, of many data, of many crop cycles, in order to apply big data approaches to be able to foresee or to predict what will be the evolution or the population dynamics of the different pests. So with this, I think I finish. Just acknowledge our founders. So Onema in France, and I cannot pronounce this in German, but probably some of you. And also I would like to remark that Fundación Cajamar, that was not eligible for the call, has been collaborating in a partnership, an important partner of the project in their own means. Okay? This is what they do, but I think it's very late. Thank you very much.
Okay, are there any questions? Quick questions, because we are a little bit out of time.
I'm just wondering if you're collaborating with insurance companies, like when you're developing the DSS. So, like the adoption of a DSS could be like linked to an insurance for the farmers, so they would be they would get some money if something happened while they're actually using the DSS.
No, we are not working with insurance companies. As I don't think the main phytopharmaceutical companies, as Bayer or whatever, are working with them. I mean, I don't really.
But I just know that some countries like Italy, for example, has some insurance linked to DSS adoption, like integrated pest management adoption, and that's something that actually motivate very much the growers.
To use it.
Yeah.
Okay, I didn't know that, but it's a very nice suggestion to try to contact. Thank you very much.
Somebody else? No. Somebody else a question? Otherwise we can, at the end of this session, and then we're going to the next speaker. The next speaker is Katharina Wechselbergen, and she's going to introduce us to the world of the wireworm. So, oh, sorry.
I try to be fast.
something?
It's not working here.
It's not working, no? Oh no, it doesn't work now.
Change the battery. No, try it.
It worked 1 minute before.
Sorry.
Oh, it's okay. Thanks. Okay. Well, one more try. Perfect. So the slide will be changed. Oh, perfect. Sorry. Okay. Good afternoon, everybody. My name is Katharina Wechselberger. I am working at the Austrian Agency for Health and Food Safety, and I'm the coordinator of the project ELAD Pro. We are 14 partners from 6 European countries working on this project, and 2 of our partners do not even get funding, so we are quite motivated to work on the topic. Well, wireworms are the soil-dwelling larvae of click beetles, and they cause severe damage in field crops, but also in vegetables. And they are one of the most difficult insect pests to control. And this is because wireworms do move in the soil throughout the vegetation period. When the conditions get unfavorable for the wireworms, they dwell into deeper soil layers. And when they are in deeper soil layers, they cannot be reached with management. For example, with mechanical approaches. So for us, it's quite important to know when they are dwelling in the upper soil layers. Those of you who have read about wireworms will know that there's a lot of information that they are dwelling twice a year in the upper soil layer, once in spring and once in autumn. But in other papers you will read that they are once in the upper soil layer in summer and once in autumn. So it's quite different, and this is why our partners in Germany decided to prepare a modeling system. And it is called SimAgrioW, and it was developed in 2012. And it is a 2-phase model, which is mainly based on soil temperature, soil moisture, and soil type. And when the model was evaluated in Western Germany, it had quite a good hit rate of about 80 to 85%. However, when we evaluated the model in a former project in Eastern Austria, we found out that we had some problems. We had a hit rate of about 54%, and on one side, the measured wireworm activity and the predictive wireworm activity were even diametrically opposed. So our colleagues in Germany didn't believe this result. However, when we decided that we have to implement more factors into the model, we knew that soil moisture and soil temperature and soil type are not enough to predict wireworm in the upper soil layer for throughout Europe. So we decided to make a continuous wireworm monitoring across Europe, and also to make semi-field trials and laboratory tests. In the laboratory, we are testing the influence of plant volatiles on wireworm behavior and the influence of soil temperature and soil moisture without any other influencing factors on the behavior of wireworms. But I won't get into detail on that. At the beginning of the project, we designed a monitoring protocol in order to make sure that we can compare the different outcomes from the different European countries. And it is, well, it's a protocol which describes that we should have 10, well, it's some kind of a bait trap. It contains germinating maize and wheat, and at least 3 times a year we also had soil sampling. And we measured soil temperature and soil moisture on the site. However, we had some problems with drought, this caused some problems with the data logger. So we will have to model the data maybe throughout the next year for some sites. Here you have the first it's from Austria. It's from 2018. It's one site with Acrotis ustulatus. It is the same species which occurred on the site mentioned before. And once again, you can see that the measured wireworm activity is diametrically opposed to the outcome of C macro W. So we proved that they behave completely different than other species which occur in Western Germany, for example, Acrotis obscurus or Acrotis scutator. But we also compared other influencing factors, for example, the soil coverage. Here you see the results of a trial site in Tyrol, in Western Austria. And here you can see that this is a trial on a site where you can find potatoes beneath a meadow. And we know that the wireworms are distributed on both sides in a similar, you find similar amounts of wireworms in both sides. And as you can see in the grassland, as you can see in the grassland, the wireworm activity in the upper soil layer is quite moderate throughout the whole vegetation period, whereas in the yellow, whereas the wireworm activity in the upper soil layer in the potato field was quite low in spring when the vegetation cover was low, and it increased when the vegetation cover increased. So we do know that vegetation cover is another influencing factor that has to be implemented into the model. Okay. Here you can see a similar trial which was conducted in Switzerland. Once again, we compared a meadow. These are the green stripes. A potato field. These are the yellow stripes, and a field of bare soil. These are the gray stripes. But I have to say that the field of bare soil wasn't bare because we had some problems with weeds. So. However, we can see that in this meadow, in this field, in the meadow, the wireworm activity changed throughout the year, but it was quite moderate. But there was an influence, so the soil moisture had an influence on the occurrence of wireworms in the upper soil layer. So it's quite complicated to have several factors in a linear model. We knew that this might be a problem. On another field in Flanders, our colleagues tested the influence of the soil management on wireworm occurrence, and we found out that the ploughing didn't have enough influence on wireworm occurrence. Sometimes we had a lot of wireworms, sometimes it was quite low. However, when they worked with the spading machine, the wireworm activity was quite low in both years. I don't know if you know these types of machines. The ploughing machine turns the soil around, and the spading machine crashes the soil. So the structure of the soil is destroyed, and it dries down quite quickly. And we think that this dry down of the soil is the most important factor. This would be another factor that would have been implemented into the model. So it's getting even more complicated. In order to have some outcomes from only one wireworm species, our colleagues from JKI prepared these semi-field trials. They had cages, and in each cage you found only one wireworm species, and it was quite interesting because they also had a field site a few hundred meters, a field trial a few hundred meters beside this cage trial. And they also had a problem because in 2018 the wireworms didn't dwell in the upper soil layers in autumn, although we knew that these species would dwell in autumn. And in the next year, in 2018, we found a lot of beetles in the cage. So we also knew that the life cycle of the wireworms have to be involved into the model. So the significance for IPM is that we have to know when wireworms dwell in the upper soil layer in order to make decisions on timing of control measures. This might be interesting for tillage measures, but also for harvesting, particularly in potatoes. Maybe you know that the damage in potatoes is done in autumn by wireworms, so sometimes only a few days difference in harvest time can make a change in the damage. Well, we are in the second year of the project, and we have to we dedicated the first 2 years of the project on data collection, and the third year will be dedicated to modeling. However, we started with our first approaches. One of the approaches is a linear system for Agridotis ustulatus, and we found a way to simulate their behavior. However, we still had problems in this new approach in periods where the temperature is below 11 degrees, as well as in periods when the temperature is the soil moisture is quite low. So some work has to be done here. Our colleagues from Egypt, from France, do have a new approach. They decided that we should work on a mechanistic landscape model. It would be easier to work on a mechanistic model because no more factors can be included compared to a linear model. And they also want to incorporate into the model the development of the beetles as well as the development of the wireworms. So you would have a combination of the whole developmental cycle. And one of the most important factors influencing the occurrence of beetles, of click beetles, is the landscape. When you do have a lot of grassland in a landscape, the potential for wireworm occurrence does increase. So they decided that we should include the emergence of beetles, as well as the oviposition, and also the movements of the wireworms in the soil column. So as you can imagine, these are a lot of data, and we will not be able to collect these data within this project, but maybe this will be the content of further projects, of future projects. Well, so in the next year, as I told you before, we will have to prepare data because of the missing climate data, because of drought and some technical problems. And we also do have to identify the wireworms because it takes quite a lot of time. We collected hundreds of wireworms, and we need to do a molecular analysis with each of them. And throughout the next year, we will try to find one type of model in which the most influencing factors, most important factors are included. So at least that we have a base for a future work. These are the partners working on the project. Our colleagues from Germany, from ESIP, do have prepared some kind of online decision support system based on SimAgriOW, and this is not freely available. It's not open access, and it's only available in German. However, they started to translate it in English, and we decided that this will be the platform where our work will be published open access at the end of the project. So, if you have any questions, it would be a pleasure to answer them. Thanks.
Cheers. May I have a question? Thank you for the nice presentation, and I think if we can offer something for farmers which somehow covers 3, 4, 5 years time period regarding the life cycle of wireworms, that is something. However, the question is the sampling intensity, because farmers, of course, they are limited in time, in money and whatever. So can you elaborate or explain a little bit the sampling intensity and then the strength of the data? I mean, number of samples per hectare, per whatever.
This is a good question. We were limited in sampling abilities ourselves. So we had 10 samples each week or every second week, and we had to analyze the amount of wireworms, and we know that no farmer will be able to do this. But on the other hand, if we would know if there's some kind of opportunity to find out if the danger for wireworm infestation is higher, it would be easier to say that the outcome of the model has to be respected when the farmer decides to choose the different crops for crop rotation, for example, or to choose which kind of control measures he will or she will implement. So it is a problem, and we have to solve it. But I think that it only will be solved when we take into account that, for example, in Austria, we didn't have any possibility this year to fight wireworms effectively. So farmer had losses of about 70%. And when they have this pressure, they will use this kind of possibility to yes.
Thank you for the presentation. I just had a question about a big.
Any other questions? Question?
Yes. I had a question about the monitoring of adults, possibly. If it was easier, or because it seems very complicated to monitor the wireworm in the soil.
Yes, it's easier to monitor the adults in theory, because we do have pheromones, but we found out that the positioning of the traps does have a large impact on the amount of catches. So we would have to prepare some kind of a monitoring guideline for the monitoring of adults too. And we didn't have at the beginning of the project, because we thought we were just monitoring wireworms. But two of our project partners are monitoring beetles too, and they found out that that they have different results with different kind of traps, and also when they are positioned at a different height. So it's not that easy. And on the other hand, it's if you do a monitoring on wireworms and on beetles, and you compare the data from one field, you will find out in many cases that the species found in the soil are not the same than those found in the traps of the beetles.
And what's the reason?
Well, the reason is because I think that the reason is that the different species have a preference in oviposition. They do like a high amount of TOC, for example, or they prefer some grassland. And when you have a pheromone trap, the male are attracted, and they come to a place where the female beetles wouldn't fly. This is the reason.
So maybe keep the questions for the general end session. Next speaker is Ingeborg Mensel-Hokkanen. I don't know if it's pronounced. Okay, somebody else. And he's going to talk about novel biosafe IPM strategies to manage pesticide resistance in pollen beetles. Okay.
So thank you very much. Indeed, I'm not Ingeborg, she's sitting there. I originally coordinated this project and started and more than halfway through. And then I was facing mandatory retirement from the University of Helsinki because of age. So we swapped and I can't represent officially the university anymore at my age. So Ingeborg has been the last, well, this year and half of the project. But we decided that I will present the current state still here. So thank you. Right, we talk about new biosafe IPM strategies to manage pesticide resistance in pollen beetles. So this targets the one aspect in the application rounds of managing pesticide resistance rather than some of the topics that were previously presented. So we are a consortium of 4 partners, leading University of Helsinki, and then other partners from Belgium, Ghent University, Aarhus University from Denmark, and Estonian University of Life Sciences. The background to this project is that the pollen beetle is a key pest in oilseed rape throughout Europe, and it has evolved over the past about 15 years. High levels of resistance to pesticides throughout Europe, to that extent that in 2008 there were crop failures in 2006, 2007, and 2008 EPPO organized an ad hoc workshop, large workshop, only for pollen beetle and what to do about the pollen beetle and its control in Europe. Now, 10 years later, late last year, EPPO organized another workshop on how to deal with oilseed rape pests, and it was sort of sad to realize that actually very little progress had been done in the past 10 years after the first workshop with about 50 people. So we are still facing this same situation. Resistance is widespread, particularly for pyrethroids, which was the only class of insecticides allowed in most countries about 10 years ago. So that obviously is the reason. And now, in the last workshop, less than a year ago, it was reported that there are some cases of reduced sensitivity of pollen beetles to neonicotinoids. So it's starting the same cycle again. So the main objective of IPM for meningitis, that's the acronym of the project, is or was to develop new, safe and sustainable, economically feasible strategies for pollen beetle control, with the expectation that we don't need to resort to insecticide use as often as before. So the basic idea is, of course, that in order not to have resistance evolution, we have to reduce the selection pressure on the pest. So we propose, and we have developed, and we have knowledge on how to deal with the pollen beetle with much less pesticide use, and that would then assure the sustainable use of the pesticides that we have available. Now, our work in IPM for Alligators project focused on about 5 topics. One is, I would say, almost the cornerstone of the whole thing, is to try to find out ways how we can buffer the cropping system against outbreaks of pests, of pollen beetle specifically, but you will see also more generically. And then specific to pollen beetle, we have been developing forecasting monitoring methods and revising the thresholds. Densities of pollen beetles in any European country have been really pretty approximate, and precise data from several countries show that we have been treating far too often, resulting in unnecessary spraying. So we felt there is need for that. We have been developing targeted precision biocontrol, and as a new tool for any pest management, RNA interference methods for the control of the pollen beetle. And then last work package we had was looking at the socioeconomics and biosafety of some of these techniques. Now I start showing the results, basically from the final end, if you wish. About 1 week ago, a special issue of the journal Arthropod Plant Interactions was made available or is available on the web. It's a special issue on the ecology and control of the pollen beetle. So much of the information or new information concerning pollen beetle management you can find in this issue. Special guest editor was Ingeborg, so credit to her for that. So please take a look. About half of the articles are open access in this issue, so you can easily read about that. Now, in the title slide, we promised some information about some basic principles that apply to this situation. You have to see that we are dealing now with a major open field crop in Europe, and we have been very frustrated by the lack of effective biocontrol methods in any open field crop. So even here, anybody who has been talking about biocontrol as part of IPM has been focusing on greenhouses, which is fine. But doing work in the open field, we also would expect that we would have some effective biocontrol, and that's our aim and dream, if you wish. So I introduced 2 new concepts, which will be generic to many cropping systems, all cropping systems, I hope, and specifically, of course, in our case, to the pollen beetle. About a year ago, oops, sorry, that was wrong. Now I try to push the right button. Yes. No. That was the wrong button, wrong way. Okay. Right. Eco-stacking. I introduced the concept about a year ago, an editorial in the Arthropod Plant Interactions journal, trying to put together all the components of ecosystem services and the beneficial functions of ecosystem services, a little bit in the same way as genetic engineers are talking about stacking genetic traits into one single plant or crop plant. You have this resistance, that, and that, and that, and tolerance to this. We want to do the same thing at the ecosystem, agroecosystem level, by bringing together from different levels of biodiversity all the beneficial components that contribute to biocontrol into this same system. That's in a nutshell about the concept of eco-stacking. You can read more about this in these articles. Another concept, generic to IPM, that we hope has been, this article was published a few months ago, summer this year. Insect pest suppressive soils. I heard here plant pathologists already referring to suppressive soils, which is a well-established concept in plant pathology, plant pathogen biocontrol. There has never been anyone talking about suppressive soils for insect pests, and Google search didn't find any citations to that, so we put it into one article published this year. I will not go more into that, the mechanisms, and the insect pest suppression relies obviously on insect pathogens and insect antagonists living in the soil, and we just have to make sure that we have enough of them, the right amount, right kinds, in the right place, and that will help us enormously into biocontrol. Now, as a result from IPM for alligators, I will only focus on one major outcome, which has to do with the buffering of ecosystems against pollen beetle outbreak. And we build this buffering in that particular case, but remembering eco-stacking, there are many other components, but this one component on the parasitoids of the pollen beetle, because we know they are extremely efficient, can very well reduce the populations much below economic threshold levels if we allow them to do that. I will show you some information on that. We have like 15 years of monitoring data from Finland, Finnish rapeseed cropping ecosystems on pollen beetle levels of parasitism by two major parasitoids, starting from 1985 and completed actually last year. On the first look, it seems to be rather random variation, as you might expect. I would like to point out that the effective level of good biocontrol is achieved at about 30 to 40% level parasitism. That is absolutely enough to keep the pest under control. So anything above about 30% on the graph there looks very good. But as you see, most of the averages are below that. Now, the variation is not random. And we analyzed this, and you can easily spot extremely rapid increases in the level of percent parasitism or by a level of biocontrol by the parasitoids in the Finnish system, until there is a dramatic drop in the population density of the parasitoids from one year to the next, from 50% down to 7% in the first case. There's another increase immediately after that, not quite as rapid, but you see a clear increasing trend, reaching well over 30–40%, until there was another drop in the population densities or levels, parasitism levels, followed by another uprise trend and another fall. And the same has happened over the last 3 years, 2015, 16, 17. 2016, 17. From 2015, a drop, 2016, 17, another increase. So you see, they were always connected, 1 year high and then following year small and low numbers. And here I would like that you focus your attention to the importance of considering not only your pest, your field, what happens in the agro ecosystem around you is, or potentially equally important or even more important for your system to function. And I would not really have thought of that, but it has turned out that the pattern of crashes in the pollen beetle parasitism levels are serial aphids. How is that? Yeah. Serial aphids, there was the years 87, 88. No aphids, a lot of aphids. In Finnish conditions, about once or twice in 10 years we have really high outbreaks of aphids, and this happened here. In that year, following year, another large aphid year, and the same here. So outbreaks of serial aphids, we have very good forecasting system. We know almost no potential for outbreaks, high outbreak. So this happens twice in a decade approximately, and always results in crashes in the population levels of pollen beetle parasitoids. So how does it work? It works so that we have a rapeseed field in a crop rotation, mainly with cereals, spring cereals in the Finnish case. The parasitoids stay and breed in the rapeseed field, and they stay over winter in that particular field and emerge from that field next year. Now, next year, the rapeseed field rotates to another field, and you have spring cereals in that particular field. You have an aphid year, you go and spray, you spray your cereal fields, you spray. The spraying of the cereal aphids in Finland coincides exactly with the emergence time of the parasitoid from that field. And this obviously kills all the parasitoids. We have a significant correlation between the severity of aphid attack and the medium and the percent parasitism on pollen beetles. We have it even correlates, the parasitism level correlates with the annual sales of dimethoate, which is the insecticide used only for that purpose. So significant correlation. No correlation for insecticide sprays in rapeseed fields. But just to assure you that parasitoids are important in determining the level of pollen beetles in the following year. Pollen beetle larvae have been counted on the x-axis, y-axis percent parasitism in previous year on the y-axis, and you see that there is a significant correlation indicating that the pollen beetle parasitoids can or do influence the levels of. We had a simulation model, showing exactly that parasitoids are very easily able to keep the levels of pollen beetle below the economic threshold. So we conclude from this that if we only allow the parasitoids or take care of the parasitoids and let them do their job, we will have no problem with the pollen beetles. And if we do, then these occasional sprays with the insecticide can be still used. I'm not talking about the other results. We have exciting results from the other work packages as well. I might comment still that we have shown the proof of concept for the RNAi possibility as a new type of bioinsecticide, if you wish. And it's possible to develop that probably into this. The transparent research perspective, principles developed here, for example, the general principles, eco-stacking and insect pest suppressive soils, we think are generally applicable to IPM. And in this particular case, for implementation and farmer update, uptake. Actually, it should be rather than uptake. Farmer uptake. Further research on the eco-stacking techniques, how to precisely do it, and demonstration trials would be needed. So I thank you for your attention here. Before I close, I would like to advertise that we organize in 20 months from now to 2020 International Congress of Entomology in Helsinki, Finland. ICE 2020, that's the large entomology meeting every 4 years. It's for the first time in Europe since 26 years, so all of you are cordially invited to come to attend the ICE 2020 meeting. We have some of these brochures on the table there. Please grab something. Some of them to take with you. And right now we are still soliciting for symposium proposals. We have large section already on IPM, but many more symposia are welcome, or any other topic on wireworms or what have you, in 20 months' time. But please submit the proposal for a symposium during this year. And then there's also a few postcards of Helsinki that remind you come to Helsinki in 2020, July 19 to 24.
So we are now coming to the last part, a general discussion. I don't know how much time we have left. Well, only 10 minutes, so maybe questions for the last speaker? Has anybody some questions for the last speaker? Okay.
Hi, thank you for a very nice presentation, and also it's very interesting, this landscape and rotational effect. And I was wondering, because it's known as well that the work of the soil, like when you rotate, manage the soil, tillage, thank you, affects it, and that's been known for a long time. So we know some things about IPM in oilseed rape, but it's nothing has been done maybe to take into account this long-term cycle and of the parasitoids and the pollen beetles. And I was wondering what's your, feeling about things that we know already and why they haven't been applied. And this is.
Well, thank you very much. I think we know already everything we need to know about what to do. There are, of course, some components of what I call eco-stacking, of how to increase the numbers of insect antagonists in the soil, etc., etc. But just to work with the parasitoids, I'm confident we know everything we need to know. It's a matter of getting the message across and having farmers actually do that. Avoiding pesticide sprays on the following crop exactly at the time when the parasitoids are emerging, I think that's one key aspect. You mentioned the non-tillage after rapeseed crop. You get 4 times as many parasitoids if you don't till instead of tilling. So I forgot to mention, a normal rapeseed field produces 20 million pollen beetles per year, per hectare. So, and you get probably about, if you have parasitoids, you might get 15 million parasitoids. They are a valuable resource that people should think in terms of money. If you go to biocontrol companies and buy 15 million parasitoids, you pay a lot.
Another question, or for the other speakers? I think we have 5 minutes left, so. No? Okay, Antoine. And I will apologize to the speakers, but I had to be a strict timekeeper, so sorry for pushing you to finish your.
I've got a question to PEMATO, Europep project. You said that you don't use the wording pest monitoring system, but ecological monitoring systems instead. And I understood that in your system, it's because you are monitoring both pests and beneficials. And I'm wondering to what extent this kind of technology, this cart, and could also monitor, capture through this imaging other components of the systems, like, I don't know, the architecture of the plant or whatever, that could also support decision support system. Have you tried to capture some other factors?
It's not currently in the project. The people involved are mainly experts in fauna, not so much in the plants. But I believe we, in GROW, we work on the... You're not in it, okay. There is a project that is looking into all the details of the plant growth and also modeling that. So actually to look at the greenhouse as an Internet of Things, so all, and with a lot of sensors in the greenhouse, that monitors also the plant growth, and that can also incorporate all the info we have about the pests and the beneficials in it. But that's something for the future. It's not yet there. For now, we just start with the fauna and not yet the plants in it.
Thank you. I also should like to raise a question that came from the PEMATO, the ecological modeling. Maybe not only towards you, but for, tomorrow I would address this question for the audience. Could you foresee an ecological modeling system for open arable fields? Yeah, that's a, why not? But I would be very in favor. So I think, well, an ecological stacking somehow is in line with my question, or my question is in line with the ecological stacking. So I think one of the future scenarios might be an ecological modeling on arable farming as well.
I do have a question for the last speaker. How do you convince cereal growers not to spray against their aphids because it's harmful for the oilseed rape management? Here I am.
Well, certainly in Finland, at least, would be the same grower, because rotates in his own fields. So yes, I mean, he's welcome to spray all his other cereal fields, except maybe not that one. Or then maybe some compromise. But yeah, well, then it would be a question of trying to have some economic calculations if it's really worthwhile. Because even in large aphid attack years, where there's some research data from Finland showing that you really have to have the timing right, you have to have the aphid density right, otherwise your spray is not cost effective. So in general, I think they should probably not spray quite as much as they like to.
My question is why Finland farmers are using dimethoate or lambda-cyhalothrin to control aphids, because now we have much more products that are selective for parasitoids. We have Perimicab, we have Plenum, we have TPK, we have products that didn't kill parasite rates.
Short answer is that most of the data is from the time when there was only dimethoate, and that was so cheap and effective that there's nothing. After that came Pyrimor, which was, I don't know, 5 times or 10 times more expensive. People wouldn't spray that. And I question that any insecticide would be safe to parasitoids. But I, of course, welcome if there's any selectivity or anything better than what is currently used. Fine.
Thank you. I have just one last question. I guess everybody's getting nervous and hungry. I was wondering if, it's a question to the audience, if IPM could get a change, could benefit from a change in perspective, instead of always looking for the enemy and monitoring enemies like pathogens and pests, to switch also to monitoring beneficials and visualize them to farmers and say, like, if you do that, you have a large impact on those beneficials, and that the farmer then can decide on what he will do on the long run. Just as a question for the audience to think of.
Just a suggestion, how the same farmer may manage the beneficial insects and the aphid in the wheat. When you apply an action threshold, you integrate the risk for the beneficial insect. So it increases the level of aphids, which define the action, the spraying, and so you may prevent to destroy them, perhaps. And it is true also that with lambda-cyhalothrin, there is less impact on beneficial insects than it was the case with dimethoate. Dimethoate was very terrible for beneficial insects.
I would just like to say that we don't call it ecological monitoring, but we monitor also beneficials. In every time we are doing a sampling tool, beneficials, and if we use beneficials, they are included in monitoring. So when they are seen, if we cannot see them, we see the effects. We are counting also for the effects of some of them, when there is parasitism, for example. So I think we are all doing that nowadays.
Sorry. This is also transported to the farmers so that they understand everything?
For sure.
Okay, but in his case, not. I guess in the case of the, what we just mentioned, with the aphids and the pollen beetles. So I guess we have many systems where you have knowledge transfer in that regard, but I guess we have quite a few systems where we don't have that. That's what I meant.
I think we can close the session. It has been a long afternoon, and so I would like to thank you again, the speakers, the chairs, and the rapporteurs. And thank you. We can now continue chatting and maybe think of this, the outcomes of a discussion. It's a bit tricky because it's so diverse. The projects where some are finished or they are just starting, so it might be a bit diverse, but that's life, and that was the life of C-IPM. Mark and Jozef. This has been the 4th time. Okay, now, just before you close, you have seen on the walls these nice posters that were, and this leaflet on the IPM. The exhibition has been prepared.